In the late 1950s, Harvard neurophysiologists David Hubel and Torsten Wiesel extended Kufler's studies of the retinal ganglion cell receptive fields, first by mapping receptive fields in the LGN, and later in the primary visual cortex, V1. Like many great discoveries in science, their work was heavily dependent on the development of new technologies. And the key to their studies was the development of a novel method for recording neuronal activity, the tungsten microelectrode. The tungsten microelectrode is basically a thin wire, insulated along its length and sharpened at its tip, down to the width on the scale of the size of a single neuron. By inserting these electrodes into the brains of living animals, Hubel and Wiesel could record the action potentials from individual neurons in the vicinity of the microelectrode tip. These are extracellular potentials being measured, since the electrode doesn't actually pierce the cell membrane, but you can nonetheless detect voltage changes from outside the cell based on the flux of ions from the extracellular space in and out of the cell. These potentials are much smaller than the action potentials measured inside the cell. They're on the order of about 100 microvolts. Hubel and Wiesel's standard experimental setup was fairly simple: an anesthetized animal, an electrode inserted into the brain, and a slide projector on which visual stimuli can be shown to the animal. In the LGN and layer 4 of primary visual cortex, they found that the visual receptive fields of cells matched the center-surround organization found in retinal ganglion cells. That is, you could stimulate them by projecting light or dark spots into the center of the field, and you could inhibit them by stimulating with the opposite kind of light. These inputs, further, were monocular, meaning that it came from either one eye or the other. So far, this wasn't so different from what they found in the retina. So you might wonder, why does the brain go to all this trouble to transmit the information from the retina, more or less unchanged, across the brain? The answer to that question lies outside layer 4. Initially, Hubel and Wiesel had trouble stimulating responses from cells outside of layer 4. They'd shine spots of light, as they'd done for layer 4 neurons, but the cells didn't really respond. The breakthrough came by serendipity. While changing the slides on their projector, they suddenly evoked a big response from a V1 neuron. At first they thought it might be a fluke, but with careful sleuthing they figured out that the simple act of changing the slides produced a faint shadow line across the projector from the edge of the slide, and it was that line that was evoking the response. If they changed the orientation of the slide, they could make the cell fire more or less, and if they changed the direction of the movement of the slide edge, they could either make the cell fire a lot or fire a little. Thus was born the idea that V1 cells, to a first approximation, serve as edge detectors. They're able to analyze local regions of space and determine the orientation of edges in those regions. Over the subsequent decades, Hubel and Wiesel refined their experiments and described a wide range of phenomenology of the response properties and spatial organization of cells in V1. While the inputs to layer 4 were monocular, in V1, binocular responses emerged for the first time, with different cells responding in different proportion to each of the two eyes. Furthermore, the relative preference for one eye or the other varied gradually across the cortical sheet. So one region of cortex might contain cells that respond to just one eye, while cells in the adjacent area respond more to both eyes. And then farther away still, the cells respond to the other eye. This regular progression of which eye dominates is known as ocular dominance. The notion that ocular dominance is organized spatially into patches across cortex is known as columnar organization, because the ocular dominance is maintained throughout the laminar depth of cortex, forming columns of cells with similar preference. Ocular dominance columns, or ODCs, in V1 were found to be organized in patchy, stripe-like structures, and these structures came to play an important role in studying development in cortex. If an animal has one of its eyes patched at birth, then the organization of ODCs changes radically, with columns for the occluded eye failing to emerge. Thus, ocular dominance has become an important paradigm for studying how cortex develops, and Hubel and Wiesel pioneered these approaches. In many ways, Hubel and Wiesel kicked off a revolution in our understanding of the functional properties of neurons in cortex, and they won a Nobel Prize for their amazing body of work. We've learned a lot more about the nature of V1 since those early experiments, and we still have a lot more to learn. But the experiments of Hubel and Wiesel were truly amazing in their depth and breadth, and for that reason, many see them as the fathers of modern cortical neuroscience.